Hello everyone and welcome. In this video we're checking out variant 3 of May-June 2020. Before starting, make sure to check out the free notes I have available for you in the comment section down below. Now let's go ahead and start with question number 1. Energy drinks contain a high concentration of carbohydrates such as glucose, which is a source of energy. Glucose is a monosaccharide which can be quickly metabolized by body cells, releasing energy during exercise. Glucose changes the color of potassium permanganate solution from purple to colorless because glucose is a reducing sugar, so it reduces potassium permanganate. The end point is when P changes is completely colorless, and you will need to prepare different concentrations of glucose solution. Record the time taken to reach the end point for glucose concentrations. Use your results to estimate the concentration of glucose in an energy drink. You're provided with materials shown in table 1.1, so you could see here all the materials. You have G solution 20% glucose, you have 50 centimeters cubed of it, you have distilled water and dilute sulfuric acid and potassium permanganate, and U, which is the energy drink. It's recommended that you wear suitable eye protection. If A or B, P come into contact with your skin, being distilled dilute sulfuric acid or potassium permanganate, wash it immediately. Now, you will need to prepare different concentrations of glucose solutions, and the lowest concentration you prepare is 12%. You'll need to prepare 10 centimeters cubed of each concentration using G and W, and table 1.2 shows how to make up the concentrations of glucose solution you will use. Decide whether each concentrations. Okay, so I can guess that this question is going to be about finding out what the concentration of the energy drink is. He's asking us to prepare 20% glucose solutions, different concentrations of glucose, and then to use, I believe, this guy over here, potassium permanganate, although we usually use Benedict's test, but we're going to use potassium permanganate to, compare the concentrations to U so that we can figure it out. Now, I'm going to read the rest, but what I would recommend doing here is practicing a lot of past papers. When you practice a lot, you get familiar with different experiments. Although you won't have the specific, results, but you're able to go through each experiment, figure out what he wants, and that's very useful to you. So prepare the concentrations of glucose solution as shown in table 1.2 in the beakers provided. Label test tubes with the concentrations of glucose prepared in step 1. Put 1 centimeter cubed of 20% glucose into the appropriately labeled test tube. Repeat step 3 with each of the other glucose concentrations. Put 1 centimeter cubed into each test tubes and shake gently. And then the reaction will start as soon as you put P into the test tubes and not stop the timer. Keep it turning continuously. Put 1 centimeter cubed into each of the test tubes and start timing. Shake gently to mix. And then measure the time taken for each test tube to reach the endpoint. As each endpoint is reached, record the time taken in the space provided for raw results and do not stop the timer. Record in AII the time taken for each test tube to reach the endpoint. If the endpoint has not been reached after 600 seconds, which is 10 minutes, do not record the time as more than 600. And then he's going to ask you to write down the concentration of U. Yes, so basically, you're going to prepare 5 different concentrations of the solution. You're going to add potassium permanganate and measure the time taken for the potassium permanganate to change from purple to colorless. And then we're going to compare these times so that we can find the concentration or approximate concentration of U. So firstly, to prepare the different concentrations here, we're not doing any dilution. In this situation, we're not doing serial dilution. What we're doing is simply we're going to use these volumes. Okay, and you need to always add up to 10 because he said here we need to have a 10 centimeter cubed solution of each. So what we're going to do is that the first time we used 10 centimeters cubed of G and 0 centimeters cubed of W, which is water. Second time we'll use, for example, 9 centimeters cubed. Use your, so here we're going to use 9, and therefore the rest is going to be 1. We don't need to write units here because it's already written in the table. And then, for example, we could use 8, and then here we could do 2, or 7, and 3. Now, 6 and 4 are written, and this is only a 2-mark question, which means these 3 are enough. Now, to prepare them, we use cross multiplication. So I know that for a concentration of 20, I have a volume of 10 for G. So now, for a concentration X, I use the volume 9. I want to look for the concentration of X, so I'm just going to cross multiply. So 10x equals to 20 times 90 times 9, which is 180. So 20 times 9 times 100 divided by 10 is 18. So x is 18%. So right here you'd write 18. Then you have 8 and 2. So same situation, 20 to 10, x to 8. So 10x equals to 160, x equals to 16%. So this would be 16, and according to this trend, this would also be 14. However, let's make sure by calculating 20, 10, x to 7. So you're going to have 140 equals 10x and x being equal to 14. Now, moving on to carry out steps 1 through 8. So we already know the steps. What we need to do now is to use the space for raw results. So we need to draw a table, but not here. Yeah, raw results here. You could write down the results just quickly, okay, as soon as you see them, because like, you know, in the experiment, everything is happening so quickly. The color change, is happening at the same time. You're measuring the color change for all the glucose concentrations at the same time, and so we don't have time to, like, you know, draw a very neat table, write down the results for each one at a time. So you can just quickly, for example, write down here 20, and then next to it the time taken. So for example, this is an example, 60 seconds, and then 18% something seconds. And then you go ahead and transfer your results here. So record your results in an appropriate table. So remember, we need to write down the glucose concentrations against the time taken, the independent variable against the dependent variable. So let's go ahead and do that. Okay. And then this guy. Here I'm gonna have the glucose against the time. So I write down here glucose. I think it was, percentage concentration of glucose. Obviously make sure to write it fully. I just don't have much space. And then the time for color change here. And don't forget your units, and then spaces for each of the concentrations. So I have 20, 18, 16, 14, and 12. And then I will write down the time here. Obviously, for example, if I got 60 seconds here, I don't write seconds with it. I just keep it as 60 because the units are up in the table. So you write down your results, and then we move on to here. Label a test tube U and put 1 centimeters cubed of U into this test tube. Okay, so yes, obviously label your test tube U and then put 1 centimeters cubed into your test tube, and then repeat step 5 through 7 using a test tube labeled U. Okay, and then so you're just going to add potassium permanganate as well as sulfuric acid onto U, and you're going to measure the time for color change. And you're going to use the results in this table to find out the concentration for U. So let's say examples of time here. Let's say it was, for example, only 15 seconds for color change here, and then obviously it should increase because the less glucose, the greater the time, the slower the reaction is, and so the more time it takes for the reaction to happen. And so let's say here it was like 30, and then here it changed to 40, and then let's say here it was 50, and then 60. Okay, obviously the results could be anything. I don't have the experiment in front of me, so I'm just giving random results. Now he's saying record the time taken for the endpoint. If the unit is not written here, you need to make sure to write it. Time for U to change color, just write down the result that you get. So let's say it was, for example, let's say it's 43. So he's saying estimate the percentage concentration of glucose in U. Okay, 43, the closest concentration would be 16%. So approximately it should be 16. And anything, for example, in between, if let's say it was between 40 and 50, you write down, for example, 15%. So just the approximation based on the time. Now, moving on to V. Describe how you would modify this procedure to give a more accurate estimate of U. This is a very common question, and you need to remember the difference between accuracy and reliability. So to get an accurate result, you want to get a result to for example, a very high degree of decimal points being written, a very specific answer, okay, a very specific concentration. Whereas if you want to get a reliable result, you just want to make sure that you keep on repeating so that your result is in the correct range and it's not just something that happened based on a mistake or anything. And so to get an accurate result, you could use a narrow range of concentrations. So instead of using 20 through 12, now that we know it's approximately by my assumptions, it is, for example, approximately 16, how about we use concentrations between for example, 15, 16, and 17 with what's called really small points, for example, 16.5, that kind of thing, to figure out exactly what the concentration is. How about we use since we're looking at color, we need to make sure to use devices. So how about we use a device to measure the color change, or how about we use a device to measure the time taken for the color change? So because there's obviously always this problem with humans using obviously us, we have this accuracy problem when it comes to measuring time change, we maybe start the timer a little off, and so this is going to alter the accuracy. So instead what we could do is we could, for example, film a video of the time taken of, for example, the test tube undergoing the color change, do one at a time, obviously, because it'll be easier this way, and have the video itself have a timer in it. And that's, I think, a really common answer for these questions with color change. And so let's go ahead and write this down. Firstly, you don't need to write a more accurate result. You need to use a narrower range of glucose concentrations, as well as repeat with and then obviously make sure to write down how many concentrations you need. For example, I would write down the entire range. So for example, a range between, for example, now I know that the concentration, now based on your answer, you write down, for example, a range of concentrations close to that answer. So for example, for me, I'll write a range of at least five concentrations between, for example, 15 through 17, and this would be 15 through 17%, so 15 percent and 15.5%, 16%, 16.5%, and 17%, right? 1, 2, 3, 4, 5. Okay, and then I would also say use a colorimeter, a digital device, to measure time change, or the time taken. So for example, you write down here, film each reaction with a a digital timer in the video. For the color change, you could also, for example, do each experiment on its own. Okay. State two difficulties of significant sources of error when carrying out step 7 here. Measure the time taken for each test tube to reach the endpoint. As each endpoint is reached, record the time taken in the space provided for our results. Well, it's given away in the step, but whenever it comes to color change, it's always the endpoint. Okay, so color change, I need you guys to remember two things. In accuracy, we need a colorimeter always, no matter the type of experiment. Just make sure to write down everything you know, even if it might not seem fitting, it probably does fit. And so use a colorimeter, and always the endpoint is going to be a problem, and therefore here we're going to go in and write down that. Okay, difficulty finding the endpoint. And so here, for example, yes, there will be a difficulty finding endpoint. It will be hard to measure the time. That's why we used here a digital device to measure the time. And film each reaction with a digital timer in the video. Do each experiment on its own. Yeah, I feel again here, another error. It's just all these things that could lead to misaccuracies. For example, we measured the time manually instead of using a device. So you could write that down. We did all the experiments at the same time. This is another source of error. Why don't you do each experiment on its own? We not use a colorimeter. Do use a colorimeter. And so write down all these, okay? Just repeat the answer here. And so do each, okay, didn't do each experiment. Okay, so we could say that they didn't do experiments separately. Now, describe how you would modify this procedure to reduce one source of error as stated here. Okay, no, yeah, okay, exactly as stated here. So how would I, for example, fix one of these two? Well, the second one is pretty easy. I could just do each experiment separately. The first one, I could use a colorimeter. So use, what he said, one source of error. So you need to choose carefully here. I feel like it's better if we use the second example. Don't do experiment separately. Just make sure to do each solution on its own. When he says 1, you need to stick to 1. Okay. B. An investigation was carried out into the effect of drinking of an energy drink on blood glucose concentrations over a period of 3 hours. The result is shown, so you have a time, and you can see how glucose concentration increases, reaches this point, and then starts to decrease again as the time moves on. So he says, plot the graph of the results. You have to understand that when plotting a graph of results in biology, it's better if we use straight lines. So if we have points, we connect them from point to point via a straight line. And second thing to know is that we don't accept feathery lines. You need to make sure your lines are straight and clear, not too dark. Okay, so don't make scratches all over the paper and have it like stain the paper on the other side. Just make them, observable so I can see them, as well as like not feathery, not broken, that kind of thing. And so firstly, in the table, we got I think half the marks are going to be on the labeling. So where exactly are you going to put the information on the x-axis or on the y-axis? Well, the independent variable is going to go on the x-axis, whereas the dependent variable goes on the y-axis. In this situation, because we're measuring glucose concentration, which, it's, nice, we usually, in the experiment before, we just had time be the dependent variable. In this situation, actually, time is the independent one, and the glucose concentration is the one we're measuring, and so that's a dependent variable. So we put glucose concentration here. We write down the units, never forget your units. So, millimole per decimeter cubed, and then time in hours on the x-axis. And then you go ahead and start accordingly. So for example, here we have 0, 0.0, 0, 0.5, 1, 2, and 3. So since we have a bunch of blocks, we can make sure to we can go ahead and skip by 0.5. So we have 0, 0.0, 0, 0.5, 1.0, 1.5, 2.0, 2.5, and 3.0, if I'm not mistaken. Yes, okay. Then I have these guys, 5.2, 7.5, 9.1, 8.4, 7.9. So they're all in the range between 5 to 8.5. So what's it called? Let's see how many blocks I have. I have 1, 2, 3, 4, 5 blocks. And so I can go from 5, 6, 7, 8, and then 9 and 10. If I skip by 1, for example, or I can go 5, 5.5, 6, 6.5, 7. I don't think that will be enough, so it will be best if I go from 5.0, 6.0, 7.0, 8.0, 9.0, and yes, well here we skipped by 1 because we just need more than half the graph to be filled, and that's exactly what we're gonna get with here, since the biggest point here is 9.1. And so 9.1 is around here, which is, yes, more than half the graph. So starting off with this first point, 0.05.2. So this is going to be right around here. Then going from 7.5 at 0.5. So 7.5 at 0.5. And then 9.1 at 1. So right here. And then we also have 8.4 at 2. 8.4 at 2. Interesting. So going here, it's right there, and then 7.9 at 3. So at 3, we're going to go all the way to 7.9, which is right here. Now, you're going to need to get your ruler, and you're going to put it right here. So basically like this. Okay, from point to point, specifically. And you're going to draw your line. And again, not too harsh. Use a sharpened pencil. Oh my god, okay. Second, like this. And again, practice helps with these kinds of things. So make sure to practice a whole lot of paper 3. I know a lot of students actually tend to skip practicing paper 3 because there isn't anything to practice. There aren't a whole lot of questions, but the questions that you can practice, they can save your grades. So even if you don't know how to draw, even if you get something wrong because you didn't label, you can still save your grade. Okay, moving on to 2. K1 is a slide of stained transverse section through a plant root. You're not expected to be familiar with the specimen. Use a sharp pencil for drawing. You are expected to draw the correct shape and proportions for different tissues. Draw a large plant diagram for the whole root. Use one ruled labeled line and label to identify the endodermis. Okay, I don't have the microscope in front of me and I can't see the slide they had for that session. But what I do know is that for such questions, I highly advise that you guys fill up your past papers. So go on the internet, find a picture of, let's say, a cross-section of a root and draw it. And how do you draw? Well, here are the tips that you need for drawing. Firstly, you need to make sure that there are no feathery lines. And this is very important everywhere. No feathery lines, no feathery lines, don't do this chicken scratch thing. Okay, no, just draw straight, okay, confident lines. And even if you feel like it doesn't resemble the root, every one of you guys is going to have a different cross section. I mean, it's basically almost the same. It's just that, you're going to look at it maybe at different orientation. The examiner will have no idea that it wasn't just curved exactly the same way. What matters for the exam is that overall it has the same layers, the same number of vascular bundles, the same main points. It's not about how much curvature is in the drawing. Now, when it comes to labeling and drawing, we don't need any shading, so no shading. Okay, even if, for example, you find that one part is stained darker, you're not shading that part. And you're going to label. Now, labeling is going to be according to the question. Here he said to label the endodermis, so you're only labeling the endodermis. Whereas if he did not ask to label, you're going to have to label the layer that you have in front of you. Okay, no matter, if he did not ask, you're just going to have to label everything. If he asked, you're just going to label the thing that he wanted and nothing else. And how you label is also very important. So make sure to label with straight lines. So if I, for example, draw a cross-section of a root, and obviously not like this, I'm definitely going to get a 0 on this drawing. But anyway, I draw this cross-section of a root. So yes, you're going to need to label with straight lines, okay, and the straight lines are going to be parallel to each other. If not parallel, just make sure there are no broken lines in labeling. Don't do your labeling all over the place. So for example, a line like this and a line like this. Your lines, firstly, again, need to be straight. So for example, I'm labeling this guy, line needs to be here. This guy, it's better that it's parallel. If you can't label on this side, go on the opposite side. Okay, let's label, for example, here. Okay, just make sure your lines are straight, mostly parallel to each other, and that's another tip for labeling. And that's it. Now I'm going to, for example, put a picture here for you guys to look at, how I would draw a specific cross section. Moving on to 2. Observe the design and vessel elements in the center on root. Again, here you're drawing cells. And again, do not shade the cell wall, do not shade the nucleus, do not shade anything. Label only what he asked you to label. And if they didn't ask you to label anything, label everything. And that's it. Moving on to B. Figure 2.1 is a figure of photomicrograph of a stained transverse section through a root of a different plant species. You're not expected to be familiar with the specimen. He's just saying prepare an appropriate table so that it's suitable for you to record the observable differences between the root on K1 and the root in in figure 2.1. Okay, so the root that you have here and the root in front of you. So record the observable differences in your table. So firstly, to draw the table, we are going to have a table which says the differences. Now, in the case it's only said compare and contrast, or just compare, he's not going to say contrast. In the case it says compare, well, compare means similarities and differences, okay? So don't just write differences. But here you specifically asked for differences. So what I'm going to do here is I'm going to write down differences here, or let's say properties here, okay? Okay. Properties. And then here I'm going to write the slide, which is K1, and figure 2.1. Now, properties, obviously this depends on your slide, but in general it could be, for example, in the thickness of the cortex, in the number of vascular bundles, in the presence of trichomes, in what else do we have here? You can also, for example, talk about the epidermis, how many layers. So again, depending on the slide in front of you. Now I'm gonna write down here, I'm gonna get the answers from the mark scheme so that we know what we're talking about right now, and I'm gonna get back. So let's see the answers from the mark scheme and talk about them together. Okay, so he said here in the properties, what he's looking for is large xylem vessels, vascular bundles, number of air spaces, size of air spaces, and shape of air spaces. Now these are 1, 2, 3, 4, 5 answers, and you have 4 marks on this question. 1 mark will be on the table, 3 marks on the properties. Make sure to write down more than 3 properties. And here he discussed the large xylem vessels. So for example, here we could see we have really big xylem vessels. Now obviously I don't have the other slide, but you're going to have to compare them. So if this is bigger, you write down here, for example, bigger, smaller, vascular bundles, again, maybe size. You can comment on size if the vascular bundles here are bigger, smaller, narrower, thinner, that kind of thing. When it comes to number of air spaces, here we do have air spaces, just not so many. Like here, this is an airspace, this is airspace. Maybe the slide had a lot of air spaces. Okay, so we can comment on that. Size of the air spaces also matters, shape of the air spaces. Anything that comes to your mind, just make sure to write it down. And now we're going to move on to figure 2.2 is a photomicrograph of the same root section as figure 2.1 with the central area enlarged. So I have cell T and I have cell Q, and he says measure the line Q and line T to find the diameter of cell Q and cell T as shown in figure 2.2. Well, sure thing. So we're gonna get the ruler, and obviously this might not be correct because I'm not exactly on the correct scale. So my answer will be different to yours, but you're going to get the ruler, you're going to measure the diameter. So here, for example, it is 2.5 centimeters, 1.5 centimeters. 1.5 centimeters, which you can't write down in centimeters. You need to make sure to record your answer in millimeters. That's going to be 15 millimeters. Okay, so again, I measured in centimeters, but then I have to convert in millimeters. Never ever write down answers in centimeters in paper 3. All your numbers are in millimeters here. And how you convert, you just multiply by 10. So centimeter times 10, that's millimeter. Diameter of cell T. Okay, so let's measure the diameter of cell T. Again, my answer will be different to yours. So let's see here. It's approximately 0.6 centimeters, which is 6 millimeters. Okay, now 6 millimeters. He's saying here, calculate the percentage difference in diameter between cell T and cell Q and show all the steps. Now the percentage difference is different to just percentage of something over something. Percentage difference, we're going to subtract the 2 and then divide that by the bigger length. Okay, so I'm going to get the 15 minus 6 over 15 times 100. And so that's going to be 60% difference. So show all your steps. Also write down the formula you used. So for example, bigger minus smaller, or Q, diameter of Q minus T divided by Q, that kind of thing. Use your knowledge of adhesion and cohesion. Suggest why the difference in diameter of cells Q and T may affect the movement of water in the xylem. Now, cell Q is bigger than cell T. And obviously, when something is bigger, there will be maybe less adhesion to the walls, since the walls are farther away, and so now there's less adhesion, which means there's less capillarity, and so maybe there is more resistance in the narrower one, because it's narrower. And so obviously, well, in physics we know that resistance increases when a wire gets smaller, and I think it works the same either way. And what else do we know? We also know that if it's narrower, there might also be, for example, more adhesion, maybe more resistance, more cohesion. Let's write that down, okay? Between water molecules in xylem of T than Q, since it's narrower, more bonds will form, and more resistance, so slower flow in T than Q, since it's also narrower. So again, slower flow and more more cohesion, more mass flow in T than Q because it's narrower. Now this is it for this video. I hope it was useful for you guys. If it was, make sure to like, share, and subscribe, and I'll see you guys in my next one.